Hello, lovely viewers watching us from the comfort of your home. You are welcome to SHSR on Joy Learning. This is a revision show from Integrated Science. I'm your facilitator, Osei Kwame Amwatin. Today, we will focus on the topic cells. And per the syllabus, other areas need to also be added up to finish the complete area of cell. So we also look at cellular organization, transport of substance across a cell. Yes, my role here today is to help us revise this basic topic. It's a concept topic to inform one. It's an area that looks simple, but most times you, the student, make a lot of mistakes. So today, I'll highlight your pitfalls and also help remove the fear that you get when it comes to integrated science. I hope you are ready for us to take the journey. At the end of our revision, you as a student and myself will be able to describe the structure and function of plant and animal cells. In addition, we're able to describe the cell arrangement and organization within an organism. Also, we also explain the process of diffusion and osmosis. I believe you are in Form 3. You've done this when you just entered your class or a few days after you entered your class. So it's been long, but we're taking you back as a revision show to help you recover or recall these topics. So let's begin with the concept of cell. I have to ask you one question. I believe you know what a vehicle is. There are many forms of vehicles. But on what grounds will you say something is a vehicle? Think about it. Let me ask you another one. At school, there are many types of buildings there. What you call the dormitory, the dining hall, the assembly hall, maybe classrooms, administration. On what grounds or what will you use to determine that these are buildings? Because you have the school field, but you don't call the school field a building. So with these two preamble, then we need to find a standard to say that this thing qualifies to be called that. For the building structures, we believe that all buildings start by putting a block on the ground. So we say that any building that has a block in it, or any structure that has a brick block, whether cement or brick, let me put it that way, will be considered as a building in school or at school. The school field doesn't have that, so you can't consider the school field a building. Back to the vehicles. What will you consider to say something is a vehicle? Is it the mode? Is it the battery, the engine, the steering wheel, the car tires, or what? I believe something basic should be there that will let us call it a vehicle. Remember, in the olden days, they will sit on a horse or a chariot and they will move. Will you call it a vehicle? In the modern sense, we will say that anything with a kind of an engine that propels the vehicle or any structure, automobile structure like that, will be classified as a vehicle. So just having an engine that can move something on four wheels at our level will be accepted. So with these two examples, everything that we give a name to should have something basic in it. If you say something is a furniture, there should be something basic in it that will qualify it as a furniture. So to our topic, there are many living organisms in this world, or there are many substances in this world, living and non-living things. For you to be classified as a living thing, there should be something basic that when we take all living things and we look into it, we can find the same thing in it. And biologists have called that thing a cell. Yes, the basic thing that you can find in anything called a living thing is called a cell. So it means that if I take a stone and I break it into pieces, once I can't find a cell, I can't classify a stone as a living thing. Same, if I take probably a polythene bag or any polythene, and I also break into pieces, shred it, and try to use a microscope to look into it and see if I get any cell in it, I won't get it. So because of that, I can't call it a living thing. But on the other side, if the thing is a living thing and you take time and look into it, you see that a certain design or pattern of something is in all living things that we now call it the cell. It doesn't care the shape, the nature, the design of it, but it has something in it that all living things share. And because all living things share that thing, we call that cell the basic unit of life. Yes. So, to define a cell, we will say the cell is the basic unit of a living thing or life. As I said, don't care about the design, the shape, or the nature. It has to get some properties or characteristics. Once it qualifies to be that, we call it a cell. To remind you again, one scientist or two scientists are usually associated with the topic cell. And our famous scientist that we always mention, his name is Robert Hooke, rather not Hawke. So, Robert Hooke, let me spell it, well, typo mistake. Robert Hooke, in 1665, used a microscope to look at the bark of a tree and saw tiny, tiny holes in them. And he called this thing a cell. The next scientist we mostly mention is Anton van Leeuwenhoek. He also used microscope like Robert Hooke, but he used it to look at a stagnant water, look deep into it, and saw tiny, tiny, tiny things in it. At that time, he called it animalculous. That means tiny animals. Over the years, he's called the father of microbiology. Now that you have an idea of what a cell is, I'll give you about 30 seconds to look at a picture that would pop up on your screen. Take your time and study it. It's the basic structure of an animal cell. And this one is taken using the electron microscope. So you see the caption there is ultrastructure of an animal cell. What it means is that if you use the normal light microscopes we have at our labs, you can't see details of what you have on your screen. So I give you 30 seconds to go around, view it well, look at the names, refresh your mind, and we'll zoom into its functions and its structure. So give you 30 seconds. Take a critical look at this, because I'm going to give another picture, and you would also have to take a critical look at it and see if you can identify some differences between this animal cell and the next one. So we'll move to the next picture. This is an ultrastructure of a plant cell. I'll also give you 30 seconds to look at it well. Okay. These are ultrastructures that requires an electron microscope, a very big microscope, to look at the details of these things. So, I'm also going to give another picture, which is a picture of what you may see under a light microscope, using the microscopes we have at school. So on your left is the animal cell, On your right is the plant cell. This is possibly what you see under a microscope. So we just caption a simplified drawing. Our syllabus requires us to mention most of these organelles, describe them, and also its functions. But before we go on, let's move from the outside of the animal cell to the inside, and let's compare it to that of the plant cell from the outside to the inside. You notice plasma membrane, which is also called cell membrane. After moving into it, you come to the cytoplasm. Then you come into the mitochondria. We'll describe all these. We'll come to the vesicles, glycogen, granules. Then we centered on the nucleus, where we showed nuclear envelope, chromatin, and nucleolus. Let's move to the plant cell. Moving from the outside to the inside, you won't go straight to the plasma membrane that we saw on the animal cell, but rather you go first to what we call a cell wall. So after the cell wall, you see that there's a plasma membrane. So as I asked, if you are very observant, you see some differences. So in the plant cell, we start with a cell wall before we go to a cell membrane. Then take your time and locate the cytoplasm we saw in the animal cell, and also locate it in the plant cell. What can you observe as differences? Look at the quantity there and look at the quantity in the plant cell. We'll use it for differences. Let's go on again. Can you identify any structure you see in the plant cell that you can't see in the animal cell? Let me help you. Let me use a green pen to underline this one, the chloroplast. In addition, you will see a big vacuole. Though there are vacuoles in the animal cell, but we didn't see any big vacuole like that. So look at it and look at a name called tonoplast. Again, can you identify another difference as a good student between the nucleus and the nucleus in the animal cell and the nucleus in the plant cell? Yes. Look at the size, look at the position. Keep this and don't forget, because the few things we mentioned will be used as examples of differences between plant and animal cell. Now let's go back to the drawing here. The reason I said look at this again is that in the exams, you will be sometimes asked to draw or to label organelles or parts of the cell that you are looking at. The trick in understanding how to label this is the shape. So take it again from me. We'll be looking at three things: the name of the structure, the shape and location, or the shape and the function. In drawing, they require you to identify this, and you can only identify it easily if you know the shapes. But in the theory, that is the section B, you'll be required mostly to answer a structure and its function issues. So take your time and look at it again. What can you say is the shape of the nucleus? Look at the nucleus, the entire nucleus. You can see the cursor going around it. We will say this is spherical. What about the mitochondria? Mostly say rod shape, or we say the sausage shape, whilst nucleus will be spherical. In the nucleus, you have a lot of things like chromatin, nuclear pore. Now, can we identify any other thing? The endoplasmic reticulum. Take your time and look at the endoplasmic reticulum and the Golgi body. Look at how it is packed. If you get to the exams, look at the endoplasmic. There's a sac, then one is connected to the other, another is connected to the other, another is connected to the other. So there's a connection or a bridge between the sacs. So let me circle that area. You see connections here. But if you look at the Golgi body, they are just sacs that are not connected. They are just sacs lying on each other. And you see small vesicles at the edges. So let this keep you and remind you in the exam hall. Also, the vacuole, it's a little bigger and empty. Sometimes you may see something close to the nucleus we call the centrioles. It's also a little bit empty, but they are tiny and small. This is what I'm talking about. So in the exams, use the shapes you know to identify the parts of the cell. Now that you've done this, let's look at the functions of the parts of the cell. You've come across the word cell membrane. Moving from the outside to the inside of the plant cell, of the animal cell, that's the first structure you meet. Moving from the outside to the inside of a plant cell, that will be the second structure you will meet. And we say that there is a flexible material or structure made up of lipid and proteins. They are in two layers, so we call it a biphospho layer. What will be its function? You are moving from the outside, there is substance trying to enter the cell. Its job is to check, regulate, or control who comes in, whether you are fit for the use of the cell or not. So in our case, we say that it controls the passage of substances into and out of a cell. So you can say that's your door, the door of your house. But we, the biologists, use one term to stand for all these things. We say semi-permeable membrane or partially permeable membrane. If you say that, it stands for that long sentence I just described. The next structure you meet after moving across the cell membrane is what we call the cytoplasm. Simple. An easy way to understand a cytoplasm is the egg of a fowl. Once you crack it, you find some albumin, what we call the albumin. It's jelly-like. Because the egg is a normal cell, in our case, we will say that the albumin is like the cytoplasm of the cell we are trying to look at in this picture. So we say it's jelly-like. Jelly means that when you tell it's fluid, it's jelly. It has about 90% water and about some 10% dissolved substances, which include some ionic elements, sometimes some gases, some granules, some stored things in there. In addition, it also houses structures that are unique with membrane-bound covers. Those special membrane-bound structures are called the organelles. So we say that the main function of a cytoplasm, as you can see under the functions, is to house the organelles, and it's also the site for chemical reactions or chemical reactions to take place there. Now, I brought these two first before I'll go on to another slide where we look at organelles in the sense of organelles. What do I mean? I just said that the cytoplasm that you are seeing on your screen the cytoplasm is a fluid, a jelly-like structure that houses a lot of organelles. So what do we mean by these organelles? This, back to our drawing here, these minute structures like the nucleus, like the Golgi body, like the centrioles, like the endoplasmic reticulum, like the mitochondria that we see on your screen are all structures that are bounded. But there are some structures that are not bounded, like food substances, which are just scattered in the cytoplasm. Once they are not bounded, we can't call them organelles. But those that are bounded, we call them organelles. And once they are also bounded, they have a specific function. Your shape, what you contain, and the enzymes or whatever reaction you undertake is different from another organelle. So we define organelles as minute membrane-bound structures in the cytoplasm that perform a specific function. Yes. On your screen too, you see a famous word called protoplasm. We define protoplasm as the living part of a cell. So at this stage, when they ask you, you can end here. If they ask you the makeup of a protoplasm, then two unique places of the cell is used as the protoplasm. The cytoplasm and the nucleus as an organelle. These two things come together to form what we call protoplasm. Again, protoplasm. It keeps coming in our exams. It's defined as the living part of the cell, which is made up of the cytoplasm and the nucleus. Now that you understand these two terminologies, let's look at this. So you see why I separated this slide from the previous one that has the cell membrane and the cytoplasm. It's because we don't call those two things the organelle. So when you go to the exams and they ask you for organelle, then these are the things that we are going to look at organelles. The things that we saw in the drawing. So back to the drawing again. All the structures within this drawing are called the organelles. So we'll take it one after the other. We'll look at the nucleus, look at the function too. So with what's on your screen, we've described it in the drawing already. Unique things about the nucleus is that it's double membraned, and the membrane is called the nuclear envelope. In addition, when you enter the nucleus, it's like a house on its own within the cell. It has its own interior fluid, like the way we have cytoplasm inside the cell. It has its own interior fluid too, which is called the nucleoplasm. In addition, it has thread-like materials. So you see something like thread, when you use the electron microscope to look at it. At one stage, you see this thread, which we call the chromatin. During another stage of the life cycle of a cell, you see that thread thing thickening up in a structure we call the chromosome. Either chromatin or chromosome, we say that it is this thing that carries genes or traits from our parents. So what we say is that the chromosome has genes on it. These genes carry traits so that your parents can pass on some characters to you. So for whoever you are, whatever character you have on your body, so that those inheritable ones were passed on from your parent to you through the chromosome. Now, in addition, when you enter the nucleus again, you see a very small, dense part of the nucleus called the nucleolus. When you stain the nucleus, that place will appear darker again. It has a lot of functions, but for integrated, let's hold it there. Let's look at the main function of the nucleus. We say that it controls all the activities of the cell. How did they do that? Scientists was able to take time to remove one organelle at a time and look at its effect on the others. And when they got to the nucleus, when they took the nucleus out, all the organelles were not able to function. And once it's not brought back in time, the cell will die. That's why we say that it controls all the vital activities of the cell or organelles. I've also mentioned that it contains the genetic material of the cell. Close to the nucleus, back to our picture, is endoplasmic reticulum. You can see endoplasmic reticulum on your screen. We have smooth and rough. So back to our slide. We said these are double membrane structures, and you saw them as sac-like. And they form like channels because once one sac is there, there is a linkage, another there's a linkage, another there's a linkage. And it spreads throughout the cell. Because of a picture, they just did it for little sections, not to cluster the cell. So with this, substances that enters this organelle are able to be transported from one part of a cell to the other. So we say that it acts as a channel through which substances are transported. Another thing it does for the cell is that once the nucleus is in the center and we have endoplasmic reticulum here, another there, another one down here, it kind of divides the cell into compartments. So the reaction can take place at the top, not affecting the down. So that it's like your house. It helps you divide your house into a kitchen, a bathroom, a hall, so that what you do at the kitchen will not spill over to the hall. When you visit the washroom, whatever happens there can also come to your bedroom. So it acts like a compartment, divide the cell into parts and allow reactions to take place. Then, as you saw, watch the two endoplasmic reticulum on your screen. The one with the smooth don't have some dots on them. Then the rough one has some dots on them. That dot is what we call ribosome. Now, this ribosome job is where proteins are made. So once the proteins are made, they pass through the endoplasmic and go to where it's supposed to be transported to. Then back to our drawing, look at the Golgi body. The Golgi body has flattened sacs with vesicles clustered at the edge. Their job is to form materials, or let's say packaging materials, around structures that's supposed to be transported outside or even within the cell. So if you make hormones or enzymes and want to transport them, they just don't come in the chemical form. They need to be covered. So it's like everybody has a job in the cell, and his job is to package these things and also sometimes repair the cell membrane. The ribosome I mentioned earlier are minute structures. They are among the SS level. It is the only organelle that doesn't have a membrane. So it says non-membrane bound. Its job is to set us as a platform where protein are made. Yes, where protein are made. You may think that when you eat animals, you've gotten the protein. No. You get the material that your body divides or breaks into pieces, and your body reassembles it based on a certain code. And that rearrangement to form a protein is done on the ribosome. So we say it's the site or the platform. Our famous mitochondria or mitochondrion. You saw the design or the shape. We say its function is to provide energy for the cell. It manufactures it, stores it, and releases it as energy for the cell. And the form in which we release this is called the ATP. Mostly we term the ATP, it's a short word for adenosine triphosphate. If in the exams they don't bring production of energy, then they will bring the powerhouse of the cell. Lysosome is a small dark spherical structure that has a lot of enzymes in it. If you eat and the food goes into your cell, it's this one that is acting as now the digestive structure of the cell. It will break the whatever comes in into pieces to the form that your cell can use. If a bacteria enters your cell and that bacteria is not part of the organelles of the cell, they will know and they will send this lysosome to come engulf and destroy it. In that sense, we are saying that the lysosome is defending our body against diseases. Sometimes too, there are bad stuffs in your house. If you have a lot of dress you are not using and it's old, what do? You throw it away. So there may be mitochondria that has finished its job. It's not worth having. This lysosome will go and clean it. And we call this one the autophagy. If a cell is dead and we may not need it, the lysosome within that same cell can burst open. And we use the word it eats the cell itself. And we say it's called autolysis. In addition, sometimes we also call it the suicide bag. It's like you're there and you kill yourself. And that's a function of the lysosome. Then vacuole. In our cell, we may have excess of fluids, excess of anything. Somebody has to collect this excess from the cell so that things can go on. In that case, they create a membrane in which we pack all these things into. You can call it a storage room. Yes, your vacuole is your storage room of the cell. So it acts as a storage room, taking excess water, excess food nutrient, excess waste, excess everything and keep it. In some organelles, in some organisms, it was this thing that is sent out as a means of excreting. In others, to keep it. The day we need substances, we go to them and tell them now we need this from the storehouse. In plants, I'm not saying animal, listen to it well. In plants, the membrane around that big vacuole we saw in the picture is called the tonoplast. Yes, the word is tonoplast. So underline it. Tonoplast. So whenever they give you a plant cell and you see a big vacuole and they give you two guidelines, one into it and one outside, know that they are trying to point to the tonoplast. If you are not smart, you won't know what to write for that one. The last three, which we would tackle it when we come back from a break. So we take a short break and come back and tackle these three.